Welcome to my channel XY Biology Academy. In this experiment, we will learn the spread plate technique, a commonly used microbiological method for inoculating microorganisms on the surface of a solid agar medium. The spread plate technique is based on the principle that a previously diluted microbial suspension is evenly distributed over the surface of a solid agar medium using a sterile L-shaped glass spreader. Materials required for this practical include the following: sterile nutrient agar plate, Bunsen burner, marker for labeling. Sterile inoculating loop, diluted culture of bacteria, sterile water, an L-shaped glass spreader, 95% ethyl alcohol, a beaker, and a turntable. Now let's perform the practical step by step. Step 1. Sterilizing the glass spreader. Take a beaker containing 95% ethyl alcohol and immerse the bent portion of the L-shaped glass spreader into the alcohol. This helps sterilize the spreader before use. Step 2. Preparing the agar plate. Take a sterile nutrient agar plate and label it properly with the required information. Place 1 drop of sterile water in the center of the agar surface. Then, using a sterile inoculating loop, transfer a loopful of the previously diluted bacterial culture into the water drop and mix it gently. Step 3. Flame sterilization. Remove the glass spreader from the alcohol and carefully pass it through the flame of the Bunsen burner. Keep the bent end of the spreader pointed downward while flaming to prevent burning alcohol from flowing toward your hand. Allow the alcohol to burn off completely, then let the spreader cool for about 10 to 15 seconds. Never use a hot spreader directly on the agar surface, as it may kill the microorganisms or damage the agar. Step 4. Spreading the inoculum. Slightly lift the lid of the petri plate and place it on the turntable. A turntable in the spread plate technique is a flat rotating platform on which the petri dish is placed. As it spins, the L-shaped glass spreader remains in contact with the agar surface, allowing the microbial suspension to spread evenly. While the plate is rotating, gently touch the cooled spreader to the agar surface and move it smoothly back and forth. Continue spreading until the microbial suspension is distributed evenly across the entire surface of the agar. Step 5. Completing the inoculation. Once the culture has been spread uniformly, replace the lid of the petri plate. Immerse the glass spreader back into the alcohol and flame sterilize it again before storing it or using it for another sample. Alternative method. If a turntable is not available, the same procedure can be performed by rotating the petri plate manually while gently moving the sterile glass spreader over the agar surface. After incubation, the microbial cells grow only on the surface of the nutrient agar, producing well-isolated colonies. These colonies can be used for identification, viable cell counting, and further microbiological studies.